Hi, everybody. Thank you for joining. We'll give it another minute or so to give our West Coasters a little more time to join. Thank you. All right. Hello, everybody. Good morning, afternoon, and evening to viewers around the world, and welcome to our third Global Reaxys User Day. My name is Randy Kenyon. I lead the Life Science Solutions sales team for North and South America at Elsevier. I have the pleasure of being your host for the second half of the Reaxys User Day. It's great to see our chemistry community and colleagues together on this platform. This event is recorded live, and we have some great speakers lined up for you today. All experts will present a variety of interesting chemistry-related cases. Before we start, let me go over a few housekeeping topics. First, yes, we'll make recordings available after this session so you can rewatch or view any presentation you may have missed. Second, on the top right corner, you can expand your screen to full view and control the volume. Also, on the bottom right, you can open the chat and display the Q&A menu. We encourage you to ask questions in there. You can easily click to go from chat to Q&A to polls to handouts. Please enter your questions in the Q&A so that we can see and answer all of them. If we can't answer a question live, we will answer it after the event. There's a full agenda available under the Sessions tab. And finally, please share your feedback after each session so that we can continue to learn and improve this event. All right, now I'd like to introduce our keynote speaker, Dr. Quentin Perron. Quentin has a PhD in organometallic chemistry from the University of Geneva. He's held several roles at UCLA and Laboratoire Elsevier before focusing on data science and chem informatics. In 2016, Quentin co-founded Iktos in France. Iktos is a company that develops AI technologies for de novo drug design, and Quentin serves as the Chief Scientific Officer. Under his leadership, Iktos has made a strong presence in the field and market. Welcome, Quentin. It's great to have you here.
Thank you very much, Randall, and thank you all. I'm very happy to be here today to share with you the view of how AI is changing the field of chemistry, and I'm very happy to be able to present you that. So, of course, I'm not alone. I have my partners in Gaston Maté and Nicolas Dohu, who are Florian experts in the pharma industry and Nicolas expert in AI. And we came with this idea to use generative AI in 2016, applied to molecules. We are 70 people. We have a footprint in US and Japan, and we put all our energy and efforts into the field of organic chemistry, medicinal chemistry using AI. We are backed up by venture company like Omnes and Ventures and DPO Farm. So I think you know by heart this type of charts, how difficult it is to find a drug, starting from a target to a preclinical candidate and ultimately to market approval. At Iktos, we are focused on this part of the whole process. How to start from a target, a biological target, to a preclinical candidate. And this is quite a difficult task. And to go from lead discovery to late-stage lead optimization, it's like solving a Rubik's Cube. And each type of move, it's like making a molecules and assessing these molecules. And we do that through iterative design, make, test, analysis cycles. This is right now the only way to make molecules and to optimize compounds. It takes time, it requires a lot of molecules, and it costs millions of euros. And usually the success rate is between 10 to 20%. So there are room for improvement for sure. So the question we can ask ourselves is how AI can help in that regard. My definition of AI is that AI is a technology which is able to make very complex prediction, like going from a text to an image, or like for ChatGPT here, asking a simple question and having a quite convincing answer. This is, prediction of words, characters to make sentence, which makes sense. So ChatGPT is quite new for the public, I would say, but similar technology has been used as well in the field of drug discovery. For different type of tasks, for instance, protein structure prediction, like what AlphaFold is doing, we have more old technology, I would say, with affinity prediction using statistical models, QSAR models. There are plenty of different type of techniques to do that now. Also with the library size, the size of the new size of the library with billions of compounds now, there are new approaches with massive virtual screening, using docking softwares. There are some active learning approaches. More recently, retrosynthesis. During many years, we thought that it was only human capable of doing retrosynthesis, but now AI, multi-calorie research, reinforcement learning, or new technology which start to provide very convincing results. And the same for molecular generation, which is kind of a heart, how to design the molecules. We always thought that only chemists can do that, but now again, like with ChatGPT, generative AI is capable of designing very nice and convincing structures. So AI is really changing the game and changing how we can apply those technology and how we can move forwards new molecules, new clinical candidates. But still, it's starting point. Those AI generate in silico, propose in silico, rank in silico, new molecules, new ideas. In all cases, whatever your AI, whatever your technology, you have to make those molecules. And the problem is that right now, whatever the company looks like this. So you have a listing of molecules, you need to go to the literatures, you need to analyze what has been done in the past, similar molecules, similar context. You need to organize your workload, order your compounds, and then go to the lab, set up your experiments, the materials, launch the reaction, monitor the reaction, do the workup, extraction, solvent evaporation, and then of course you need to purify those compounds. So right now this process has not evolved a lot. Of course, at some point you have some technology which can automate some tasks, like now purification usually are done like this, not with a column and silica gel you do by yourself. So there are some minor improvement, but overall the process is highly manual. And at Iktos, we are convinced that the future of chemistry, the future of chemistry is passing by not only AI and this type of fantastic technology, but also on a new way to make those compounds. So the question and the challenge is, how can I go from a listing of molecules I want to make to powders ready to be tested, and of course, as fast as possible? So the obvious answer is automated chemistry, is robots. And it seems that we are not alone in that regard. And there are plenty of companies developing platform to automate this old-fashioned chemistry. And there are different types of initiatives, as I said, some with AI and some without AI. And I think it's very important to show you today that With AI, it's much more, I would say, optimal. So to build such a platform, of course, you have key components. You have, from your listing of molecules you want to make, first, you need to find the synthetic pathways. Then to order your compounds and, of course, receive it, manage it. Then you need a robot to execute your chemistry and do purification analysis to get the powder. But there are dependencies. So if you have a generative AI which generate molecules, and for all of those molecules, you are not able to find a synthetic pathway, forget about your platform. If you have molecules with synthetic pathway available, but this chemistry is not compatible with your robot, you forget about the platform. So you need to solve those kind of dependencies in order to make this platform viable. So you need to build a system where the design is connected to the make with all the underlying dependencies. And this is where AI entered into the game, because you have only two ways to do that. Either you target very simple chemistry, like library design. You have one starting point, one exit vector, let's say bromo derivatives, and you want to do on this bromo derivative plenty of Suzuki coupling, so the same reaction with similar building blocks, all boronate in this position. This is a good way, and in that case, you don't really need AI. You have your rack, your vials. You put A with the bromine, and then you put in each vials the boronate. But the problem of that, of course, you don't have AI, but it's simple molecules, only one step. And usually you do that manually. So it works on one series and one project at a time. So this is what I call automatic chemistry. With AI, you can do much more than that. And you can start building an autonomous platform where you have a listing of molecules to make. They are all different. You want to run different reaction at different position using different building blocks. And if you want to do that, which is more drug design rather than library design, you need AI. AI is mandatory here because you will generate and make more complex molecules. You need several reaction steps. And of course, it's a plenty different type of molecules. So it can be run for different projects at the same time. And this is the goal of the platform I'm going to show you today. So this is a platform we have built at Iktos. So we have the generation is done by Machia. Then we identify synthetic pathway using Spire. We organize the workload using Ilaka, and we communicate to the robot all the procedures. And then we externalize right now to external partners, purification, analysis, and testing. And this is something we're going to integrate in the future. So the platform is only 1 year old. So we are doing things step by step. Let's focus on generate. Machia, it's a very simple interface, very nice user interface, where you can take advantage of 3D ligand-based, 3D structure-based fitness functions, scoring function. You can import your data, build statistical models, use generative AI to generate molecules, maximizing all those objectives simultaneously. I will show you how we take care of synthetic feasibility by designing our generative AI. And if you want to take advantage of external fitness function you may have in-house, you can use our API gateway connector. So, which makes us unique on the market is how we generate molecules. And like a chemist, Machia generate molecules by leveraging organic chemistry, synthetic transformation, and commercial building blocks. Let's take an example here. In Machia, we have 1 million building blocks, and we have a neural network which predict which building blocks to use. And let's say here the top probability is this one, building block X, which is this building block, for instance. Then we have another AI which predict among 2,000 name reactions, so more than 90,000 transformations, synthetic transformation, or the chemistry you can find into patents, which try to predict which reaction can operate on these molecules. If I take the top probability reaction here, it's a bug detection. Then the system trigger again the first deep neural network to predict another time which building blocks can react in this position, for instance, this one. And given those two building blocks, the second AI predict which reaction can occur here to make that molecules. This is done virtually in silico. In milliseconds, we generate molecules like this, combining building blocks and reaction. And all those decisions, which building blocks, which reaction are taken by the AI. And we can decide to stop or to continue to grow even the molecules bigger. So as you understand, reaction prediction, commercial building block prediction, talk to each other and generate 1,000 molecules per steps. Then those 1,000 molecules will be scored using 3D ligand-based, structure-based, QSAR model descriptors, presence, absence of substitutes. This is here where the chemist puts its knowledge. And then we have a dot, I would say a value of this batch, an average, a max value. And then we feed back this information to our AI here using reinforcement learning. And the system, based on the results here, will decide to change the weight of the reaction prediction, the weight of the commercial building block prediction, to generate a new batch of 1,000 molecules. And step after step, the system learn and increase the fitness function until it reach the level of 1, meaning that we maximize all the endpoints of the target product profile. And contrary to other companies at Iktos, we have only one generative AI, but it covers all the use cases because we have incorporated all the chemistry which has been published into the patents. So basically, we can grow, we can link, we can cyclize, heterocyclization, macrocyclization. And if you do the math, 1,000 building blocks over 4 steps, 2,000 possible name reaction, you end up to a chemical space which is huge. And all those molecules are feasible by design, novel, diverse. So the output of Micah basically is a listing of molecules. For each of those molecules, we can see the decision taken by the AI to produce that molecules. And of course, we can see as well how this molecule is docked or scored over all the scores you have decided to set up to the system. So I hope I've been able to convince you that we generate molecules which are feasible, but still we need to find the synthetic pathway. And for that, we have designed Spire. Spire is our retrosynthesis technology engine. We work on that technology since now 6 years. And we have different type of interface. We have Spire API to trigger, to calculate a score, to identify routes. And we have Spire GUI to interact with the software. So I have this list of molecules coming from Micah. And for each of those molecules, I know that they are feasible. But I need Spire to identify all the possible routes to make the molecule 1. I need to do the same for the molecule 2, all the possible routes, etc. So the output of that is different strategies, different routes per molecule. Then, given that information, we send that to the scheduler, ELACA. So the goal of ELACA is to identify the best routes for each molecules in order to organize the workload. For instance, here, ELACA prefer to use the route 4 rather than 3, 2, or 1 because it's a Mitsunobu coupling which reacts at room temperature during 8 hours. And I can put this route 4 with the route X here on the same day, on the same rack, because they share similar temperatures and similar duration. So the goal of ELACA is to identify the best combination of routes to optimize the robot capacity. And then we send that to the chemist. This is a decision taken by ELACA to run the campaign. And the chemist can say, yes, it's good for batching, and it's good for chemistry, or yes, it's good for batching, but I disagree with this route, I prefer this one, and can say yes or not in the output of ELACA. And then once the routes are validated, we can order the building blocks and put the reaction in queue. Then, of course, we go to synthesis. As you can see here, we have decided to move forward with ChemSpeed. They propose robots with very reliable robots very small footprints, and they can run more than 100 reactions a day. So it's quite efficient in terms of surface and capacity of chemistry. And we have designed those robots according to our own workflow and pipeline. This is a layout of the robots. Of course, this type of robots are doing everything a chemist needs to do. So they can dispense solid, liquid, they can heat, cool down, they can stir, shake, evaporate, filtrate, etc. I would say basic chemistry manipulation. But also they can do inventorization, they can hold, move vials, read barcodes, and perform some database operation. So this is where we put the starting material. If you want to run a reaction A plus B gives C, you put your A and B here. Then we have solvent. We add solvent. We mix A and B into the reactor here. Here we have solid dispenser. We can put the base, the catalyst, etc. And then once the reaction is over, we go to the support phase extraction part of that robot in order to perform the workup. And then we take the filtrate back up here in barcoded vials, and then we go to Genevac to evaporate the solvent. So if you want to run chemistry on these robots, you need to avoid certain type of chemistry. We decided to avoid very low temperatures chemistry. We decided to avoid very, I would say, reactive chemistry like terbutyl lithium, for instance. We wanted to avoid also chemistry which generate different type of radioisomers. We prefer to buy the starting material containing the bromo, the ortho position, rather than relying on a Friedel-Crafts where you can have some radioisomerization. And of course, we will avoid some kind of very hygroscopic starting material. So to do so, you need to mine the literatures and to identify for each of the classic name reaction which procedures you want to use into the robot, because you cannot do that automatically. Of course, we have developed solution to extract automatically reaction prediction, condition using AI technologies, and this is working quite well. But the drawback of that is sometimes in the literatures, the proposed procedures is quite different from what a robot can do. So we have decided to do that on our own and to start developing our own chemistry compatible with the platform. And we have also developed in-house ways to use support phase chemistry, which is quite useful. So right now we master, I would say, into the robot more than 40 name reaction, which cover more or less 40% of the patent literatures. And we cover different class of chemistry. So the way we do that is just we take the most popular reaction into the patent and we start developing one each after the other one. And so we have protection, deprotection, plenty of palladium coupling reaction, amine reactivity, etc., etc. So now we have a robot, we have Micah, Spire, Ilaka. What happens if I want to run a campaign? So here, for instance, I have 3 projects, project 1, 2, and 3. Different type of scaffold, different type of exit vector, different type of chemistry. On this bromine here, I can do bromo-Suzuki coupling or bromo-Buchwald. On this nitrogen, I can do amide bond formation or sulfonamide, etc., etc. So this listing of 96 molecules across 3 different type of projects goes to Micah. For each of those molecules, we try to find all the routes, and then it goes to Ilaka, and Ilaka say, okay, let's go for this type of reaction. And then the goal of Ilaka, as I said, is to batch those reactions together. So the output of Ilaka is the following. Reactor 1, you can run the bromo-Suzuki coupling, and the bromo-Buchwald, and the SN2 alkylation, because they are all run at 80 degrees for 10 hours. And in parallel of that, in the reactor 2, you can run the amide coupling, sulfonamide coupling, and mitsonobu because they are run at room temperature for 16 hours. So this is the goal of Ilaka, to organize a workload. And as you can see here on those 2 reactors, we can do different type of project and different type of chemistry. This is something which is impossible to do if you don't have access to the radiosynthesis technology and AI. So then how does it work? We have Ilaka here, where we have our templates of reaction, where we have the campaign we want to launch. Again, inside Ilaka, we can do two things: either create new reactions, so to use a platform like high throughput experiment platform, and to launch campaign as well. So then it goes to a database which is based in the cloud, and we send to this database all the constraints of the routes we found. So which reaction, which building blocks, which barcodes, etc. And then the ChemSpeed here read this database to catch all this information and to start running the chemistry, to start running the robot. Of course, it works in both ways. Each time the robot is performing something, it's sent back to this database, and this database can be read back as well. So this is a way we have found to communicate easily with the robots, passing by very nice and easy to use Ilaka interface. And then, of course, all those reactions are stored into our Dotmatics ELN to, of course, to follow the chemistry. Okay, so as I say, the platform is 1 year old, so it's quite new. And during this year, we had to resave the robot, to plug everything around the robot, to connect the robot to all the API we have and all the technology we have to develop Ilaka, etc., etc. But since then, we have been able, first, of course, to install the robot, to use it on a daily basis. And now we have completed more than 6 DMTA cycles. We have run more than 1,000 reactions using different 40-name reactions, obtained 150 pure compounds, which has been tested as well. So to give you an idea, the DMTA cycle right now is 2 months, so I would say it's market practice. There are plenty of ways to accelerate that, but at least it's automated. So the design, again, is done in Makia. It takes, let's say, a week. Spaya, Ilaka, identifying routes, organize the scheduling, ordering the starting material, receiving the starting material takes 2 to 3 weeks, depending. Then the make 1 to 2 weeks, purification 1 to 2 weeks, and then biological assay. So overall, in all the DMTA, in average, we have a 60% success rate from in silico design to the powder on the bench ready to be tested. So we are currently using this platform for in-house pipeline on 3 out of 4 projects we are moving forward internally. As you understood, AI-driven retrosynthesis is a key element of the platform. It allows you to go from a listing of molecules to routes ready to be connected to an external system. So it's a key element of the platform. And everybody in the field of chemistry knows Elsevier, Reaxys, and we have decided to team up to use the best database of the world from Reaxys with the best retrosynthesis AI from Iktos to build the best retrosynthesis technology of the market through the Reaxys platform. So now you have access to that, and in the backend of Reaxys retrosynthesis software is run by Iktos technology. Of course, more is coming. This partnership is quite recent. We are already working on the next steps. So proposing this technology as API to be able for you to connect it to external system. But also we are working on the forward reaction prediction, given the reaction condition, what's going to be the output of my reaction, the side product, etc. Also predicting the reaction condition given the starting material and the product, but also Reaxys has a lot of data, particularly as well in the field of ADMET, and why not building some sort of models? And we have plenty of ideas and we are working on it right now. So I want to thank you for your kind attention, but also I want to thank our team, which is composed of computational chemists, cloud engineers, developers, AI, researchers, So talented team to develop quite new and efficient technology for new drug design. And thank you all. Happy to take questions.
Thank you, Quentin. That was a great talk. Well, I have a few questions for you, but it looks like we have several in the chat. So let's start with those. The first one, as today's world become more technology driven, especially with the rise of AI, how would you advise undergraduate students studying chemistry to prepare themselves for this shift? Also, as a current chemistry undergraduate, I'm curious regarding the changes in the field. What would be some of the important skill sets in this new age?
Yeah, it's a very interesting questions. I would say that a few years ago, I would advise you to learn Python and to learn how to code. But again, AI is making a lot of progress, and now there are some AI which code automatically. I think I would advise to be more curious about what AI is capable of and trying to browse all the available technology out there in order to be able to understand what's going to be the next revolution in that field, to be prepared actually, and to organize yourself, your knowledge consequently. So just be curious and just follow what happened, because you should not be frightened by AI, but to take it as a tool, as a new technology. And I think the future is bright, and there are plenty of new technology there. You will always still need chemists, so it's very important to learn your chemistry, your organic chemistry. Everybody is using laptop, etc., and it doesn't prevent you to still know how to count. So it's the same in chemistry. Those technology are here to support you, to help you. Just be curious.
I appreciate that answer, because as we're transitioning into this age, it allows all of us, regardless of our specific expertise, to be part of the journey. So thank you for that answer, Quentin. Next question. How many types of reactions and what kind of chemistry can the robot handle? Is the robot aware of cutting-edge methodologies, for example, photochemistry, electrochemistry, and more SP3 functionalization methodologies?
Yeah, so several answers to that. First, as I explained, you need not only to have a robot which can run this type of chemistry and can propose some photochemistry device. I'm not sure about electrochemistry, but there are some hardware which allow you to do that. The question is the softwares. You need as well to have a retrosynthesis technology which knows this type of chemistry, because again, everything is connected together. So right now we are using patent literatures. So if this type of chemistry has been published into the patent, we will use it. If not, we will not use it. So again, it's a connection between the AI and the knowledge of the AI and the hardware. So as well, as you understood, the goal is not really to perform, maybe in the future, but not right now, to perform very complex chemistry. Because we rely already on very complex building blocks. We order building blocks which maybe involve some UV chemistry, electrochemistry, because we buy already complex building blocks, but we try to assemble them using classic chemistry. So it doesn't mean that the robot is not doing electrochemistry, that the product at the end of the day, at some point, electrochemistry has been involved to introduce very interesting motifs. So again, using classic chemistry doesn't prevent you to have very original and novel molecule at the end.
Great, thank you. Third question for you. What are you doing with the chemical data that are generated? In other words, how are you storing the data back into the ELN or keeping track of the reaction to outcome results in SPIA? And then the last part of that question is, what do you do if the reaction failed and you did not get your product?
Okay, so of course we track everything and we keep everything in database, because the goal of this platform is not only to produce molecules, but to catch data as well. As I said, our vision is to start building an autonomous lab. So right now, there are plenty of decisions which are taken by human, but in the future, we want to take those decisions, at least part of those decisions, using machine learning models. For instance, purification, right now it's done with external providers, but we'll incorporate soon this into the platform, and we'll be able to predict in the future which column can be used for which compound in order to trigger automatically the purification setup given the product. Regarding here, so all this data is kept, and in the futures, we will start building model on it for different type of tasks. Then regarding the fact that sometimes, of course, there are some reactions we fail. But the philosophy of the platform is a bit different from the classic philosophy of medchem. In the medchem, usually you design molecules and you want to make it anyway. So you will try all the way, you will change the chemistry until you find a way to make your compound. Here, we do not plan to change the chemistry. We set up robust condition to do Suzuki, whatever reaction, and then we just run the reaction. The yield does not matter. If we have a low yield, but we have the product, fine. If we don't have the product, that's not a problem because we are not targeting only one molecule. We are targeting maybe 30, 40 molecules into a certain chemical space. So if out of those 30 molecules, we are able to make only 20, that's fine. We catch data points. And from those 20 data points, we can still train models and do another DMTA cycle. We'll not lose time to try to make those compounds because we want to catch data. And if the AI at the second DMTA cycle is coming back again with similar ideas where the reaction failed, in that case, we ask ourselves, okay, should we try to make this compound anyway? So maybe we'll go to external CROs and partners, or should we maybe modify the Suzuki coupling? Given this context, we should do a Suzuki V2, and this is what we are doing right now. We have different type of Suzuki, Mitsunobu coupling, etc., given the context and the molecules, electron rich, electron poor, injured, etc., etc. And this system is going to improve itself over the time.
That's great. We have time for one more question. And as I mentioned earlier, any questions that we have not answered, we will absolutely answer following the event. So Quentin, does the robot system help with purification?
So right now, as I said, the purification is handled to external providers. So once the reaction is over, we evaporate the solvents, we seal the vial, and we send it to an external provider, which performs purification and send back the pure compound. But of course, we are starting our Series B right now, and this is into the plan to grow this platform, to introduce purification, analysis, and early ADME as well, to close the loop and to go as fast as possible over all those steps.
Thank you so much, Quentin. Appreciate your presentation. I'm sure our audience appreciates your presentation and answering the questions that came forth. It's a pleasure. I'm excited for this journey that we're partnering together in. And thank you again for your time.
Thank you
for
so
the
much.
Audience, you will be automatically transitioned to the next session, which is scheduled to begin at 11:40 a.m. Eastern Time or 8:40 a.m. Pacific Time. Thank you.